Okay. Sorry about that, guys. I just pushed the wrong button. So now everyone should hear me, and it should be live. I think. Okay. Yeah. All right, we're good. Sorry about that. Yeah, if you guys have questions, let me know. And I've been sitting here talking to myself for like the last 5 minutes. Yeah, 'cause that's what I do. It's funny that I can't even chat in my own thing. Oh, hello. Can you guys hear me okay? Or I feel like there's something going on. There's something wrong, maybe. Or maybe just no one has any questions. This unit's just not too bad. I don't know. And I'm fine with that. Maybe you have questions about your lab report that's due tomorrow night. There's a little echo. I don't know what that's from. Could be my microphone is really sensitive. But okay, here we go. There's some nice questions. Okay, how important is knowing each phase of the cell cycle? I would just know what goes on in the phases of the cell cycle, just generally. Um, they're not too hard. So, yeah, and I can explain all of those things again. Good. There we go. There are some questions. All right, so the cell phase of the cell cycle, just general overview of each one will be helpful, and kind of knowing where they are on the this little handy dandy chart here. So like this chart is just helpful for understanding them. Like the order, like what would a cell need to do if it was brand new? It would need to grow. Then it's going to replicate its DNA. Then it's going to grow and prepare for mitosis. And then it's going to do mitosis. Remember, mitosis, division of the nucleus. So in order to divide the nucleus, well, the nucleus has to disappear. Chromosomes have to form. They have to separate. And the nucleus has to reappear. And then the cell has to split in two. Now you have to assign names to all those phases. But that's essentially what's going on. Right. So yeah, and there's checkpoints, just kind of bringing that in here in each one of these. So you have so here's the beginning of the cell cycle. Oh yeah, I tried to do that last time and I got punished. All right. So there's the beginning of the cell cycle, which is right here, right? And so G1S, G2. And the G1 checkpoint is the cell big enough? Is there enough nutrients? All that kind of stuff. Is there any problems in the DNA? And what this is showing, if it doesn't meet that, maybe the cell goes to G0, goes back in the G1. Maybe the cell stays in G1 for the rest of its days. Maybe the cell undergoes apoptosis. Whatever the case. If it doesn't, S-phase DNA replicates. G2 checkpoint is the DNA completed? And is the DNA replication completed? Are there any errors? That sort of thing. And then you have the spindle checkpoint, or the M spindle checkpoint, which is, I don't have a picture of it. This is the closest thing that I have to it. It doesn't really matter. Are all of the chromosomes correctly connected so that they can be separated? The sister chromatids can separate. Signal transduction. What do you have to know about that? First of all, let's go to this simple picture right here. It's 3 stages, right? Reception, transduction, response. Reception is just literally very simple, right? It receives the signal molecule. Transduction is the relay of between reception and response, and it can happen in any number of steps. And then response is some sort of cellular response, typically like the production of a protein, right? Or it could be something else, but it's probably going to be that. And so that is a general overview. You're typically going to have to apply it to something like this, right? And so this is a very common pathway, epinephrine pathway. You do not need to know the epinephrine pathway, meaning the steps of it. You just need to understand those 3: signal, transduction, response. So right, here's the epinephrine, which is, in this case, is the ligand or the signal. It is received on the epinephrine receptor, right? It is an epinephrine receptor. It's nothing else. It can only receive epinephrine. And that right there is just reception. Everything between here and response, which in this case response is release the glycogen stores, right? And so everything between there is transduction. And so this epinephrine joining with this receptor activates the receptor, which activates adenylyl cyclase, which activates cyclic AMP, which is a secondary messenger, and which turns on some sort of molecule, glycogen phosphorylase, which is going to tell glycogen to break apart into individual glucoses. When it is turned off, then those glycogens are no longer being broken apart, right? Yeah, I can do that, 4.4 and 4.5 for sure. I think a lot of people miss 4.5, and so it's kind of different than the rest in a way. So I will go over that. So that's signal transduction in a general overview. The kinds of questions that you'll be asked are like, researchers applied a chemical that reduced or that inhibited the activity of cyclic AMP. What effect might this have on the cell? Or it's going to be something like that. And so then what you have to do in your mind is you have to eliminate that cyclic AMP. I'm drawing on my finger. You have to eliminate that cyclic AMP, and then what comes out of that? Well, then glycogen is not broken up and glucose is not released into the cell. That's how you have to think about it. That's a lot of you all get tripped up on that language, and you just have to kind of look at it as a series of steps. And if you take out that step, what happens as a result of that, right? I don't know if that helps. And the signal cascade is just, I think it's on this maybe. I think it was on the other one actually. A cascade is just where a single receptor or single signal molecule is amplified, keyword, into multiple cellular responses. That's all that is. So something might be identified as a cascade, and that'll trip off in your mind that signal is being amplified. Yeah, G1, G2 phases. Go over those real quick. So the difference is just their proximity to mitosis, right? G1, mitosis just finished, and so the thing that's happening is growth, preparing for DNA synthesis. And in G2, mitosis is getting ready to happen, and so you're preparing for it. Still growing, but preparing for mitosis. G1 is mitosis just finished. Don't forget that S is in between G1 and G2. So 4.4, two main ideas from 4.4. And they were that there's ways to change the signal transduction pathway. The first one is changing the DNA or a mutation. If you change a protein, or you change the DNA, you change a protein. If you change a protein, you change some sort of thing, right? Here's a normal where these hexagon bullets go and fit into this triangle, and then these things happen, and this nucleus gets to respond. If you change the DNA, right, if you have a mutation, then you change the shape of this receptor, then hexagon bullets can no longer be received, then XYZ don't happen, then there's no cellular response. And so that is the idea there. The second idea that we talked about in 4.4 was just chemical blockers. Same concept, right? Here's normal. These little dots are chemicals. These little dot receivers are receiving them. Everything's good. Over here, the dots aren't allowed to be released. Why aren't the dots allowed to be released? It's because they're being blocked by some sort of chemical. And so these receptors never receive the dots, and so the target cell never activates. And that was the big thing here. Changes in the transduction pathway, mutation, chemical blockers can cause that. Talked about oxygen, or talked about carbon monoxide taking the place of oxygen. That was the big idea there. 4.5, we talked about two feedback mechanisms. So feedback mechanism is a way to maintain some sort of homeostatic function, which is essentially reception, transduction, response, right? In a big sense, like a whole body is doing that. Your brain receives something, it does a series of relays, and then there's a gland somewhere that responds. Two main feedback mechanisms. Negative feedback, where the product of a process shuts down the process. What does that mean? We use body temperature as an example. So you walk outside and it's cold. Right now it's cold outside, right? And your body has to maintain 98.6. It's cold. You walk outside, that 98.6 turns to 98.1, right? The body starts to shiver, because it says we can't go to 98.1 for very long without something wrong happening. And so you shiver, which is a process, right? The product of that process is to increase the body temperature. And so then the process shuts down. So the product is increased body temperature, the process is shivering, and that shivering shuts down which creates body heat. That's the product, and that shuts down the need to shiver in the first place. You could do that with sweating. You could do that with blood sugar. If your blood sugar gets high, body releases insulin. Insulin is the process, right? It causes the cells to pick up all the glucose out of the blood, which lowers the blood sugar. That's the product, which causes the body to stop producing insulin. That's a negative. Big thing for negative is something is being turned off because of the process. Positive feedback is where the process or the product of the process amplifies the process. So what does that mean? It means that, I use this example of cattle stampeding. A cow gets spooked, it takes off running. Another cow sees that cow running and starts to panic, and it runs too. Another cow sees those two cows running, it gets panicked, starts to run, and eventually it just kind of cascades into like this stampede. So the panic is causing the running, which is causing the panic, which is causing the running, right? I gave a couple of examples of this for real life. One of them was nursing. So milk production is stimulated by a hormone called prolactin, and prolactin causes milk to be produced. The baby nurses, which causes the production of prolactin, which causes the milk to be produced, which causes the baby to nurse, which causes prolactin to be released, so it's like a positive. Everything is turning on the process more and more. Then I talked about fruit ripening, and I talked about, like you get a piece of fruit and it's not quite ripe, and so you put it in a paper bag. And there's a gas called ethylene that causes the fruit to ripen, and ripening fruit releases ethylene, which causes the fruit to ripen, which causes the fruit to release ethylene, which causes the fruit to ripen. And so it's a feedback loop that amplifies or intensifies the product. And that was all that was. Two different feedback loops, positive, negative. I think I skipped one up here talking about cancer again, maybe. Yeah. All right. Yeah. Cancer is essentially when there is an unregulated cell division, right? So the cell is unable to, say, stop dividing. Remember, we talked about how the presence of cyclin in the cell, for instance, ebbs and flows, and as cyclin production increases, the cell starts to divide, and when cyclin goes away, the cell stops dividing. And so I gave the example in class that if cyclin was always in the cell would always divide, right? And so if there was something that kept cyclin from degrading, that would cause a cell to continue to divide, right? And so the cell has some mechanisms against this, like apoptosis, and it causes the cell to program cell death and essentially kill itself off to prevent problems. If a cell is unable to do that because of damage to the DNA, that's what cancer is. Cancer is the cell's inability to control its dividing or any problems that come from that. And it has to do with mutations in the DNA every time. Kinase. Yeah. Kinase and phosphatase, maybe. Is this one? Yeah, I think so. This is maybe what you're talking about. A kinase is an activator, and a phosphatase is a deactivator. Removes a phosphate, whereas kinase usually adds one that causes it to be activated. And speaking of kinase, the cell cycle has one, right? It's CDK, cyclin-dependent kinase. And so when cyclin comes in contact with CDK, it activates it and becomes something more than it was before. It becomes MPF, right? And that's an example of a kinase that activates. Oh, that's CD. Yes. Okay. Yeah, this is what you're talking about then. So yeah, I can go over this whole thing. I think is important. So cyclin is a protein that regulates cell cycle. The presence of it causes the cell cycle to happen. When you take it away, the cell cycle stops happening, or the cell division stops happening. Be more specific. And so CDK is an enzyme, right, that stays in the cell. And so here's G1, here's the beginning. CDK is always in the cell, but as the cell cycle progresses, cyclin starts to accumulate. You see that on this picture, right? Cell cycles are starting to accumulate, or cyclin's starting to accumulate as the cell cycle progresses, right? And once it gets to a certain point, CDK and cyclin start to join together. When they do, they form something called MPF, maturation promotion factor, which, shockingly enough, promotes the maturation of the cell. In this case, maturation is moving along the cell cycle. All right, so getting past G2 checkpoint, mitosis is going to occur, and this is bringing it to the point of metaphase, anaphase, that spindle checkpoint that we talked about. And once that happens, Once all the spindle fibers are connected at the centromeres, pulls and starts to pull the chromosomes apart, right? That is when cyclin starts to break down. And you see that on this picture right here, right? Cyclin breaks down, which is going to, in turn, counteract the end of the cell cycle. Hopefully that was helpful. To me, that little thing is like the most complicated part of the unit, and it's not complicated. So I don't think this unit's that bad, but I think where people get tripped up in this unit is the like some of the pictures, some of this stuff. And I would talk about the FRQ, but some people still haven't taken it. But that FRQ is a perfect example of how it's easy to get the language backwards as you guys are thinking about it. So, and I have some con questions here that I could, I guess, put up on the board. We could do. So if y'all wanted to do that. All right. This is just from Khan Academy. Some of you guys may have already done this. I don't know. We can go over it together regardless. All right. So which of the following scenarios describes a positive feedback mechanism? So again, before I start searching A, B, C, D, E, well, there's no E. Before I start searching A, B, C, D, I'm going to rehearse in my mind what is a positive feedback mechanism. It is where the process and the product keep turning each other on. Yes, correct, Nick Bell. That is correct. Cycling is necessary for the cell cycle to work. Without it, the cell cycle stops. Yes. Well, cell division stops, right? The cycle continues, but specifically cell division happens because of the buildup of cyclin. And that's why if cyclin was always in the cell would never stop dividing. All right. So which of the following is a positive feedback mechanism? In my head, I'm rehearsing what is that before I start reading. I don't want these answers to tell me what positive feedback is. I'm going to tell myself what positive feedback is, and then I'm going to go find that in the answer. That's how you do it. You do the opposite. You let the test trick you. I'm not going to let the test trick me because I know what positive feedback is. All right. Several hours after a meal, blood glucose levels begin to drop, prompting the pancreas to release secrete glucagon. Glucagon convert liver stores to glucose, raising blood glucose levels. Nope. It's not, because if you raise blood glucose levels, then what's going to stop happening? Glucagon production. So that's negative. During exercise, increased body temperature causes the body to sweat. Sweat vaporizes, cools down the body. Again, negative. After eating, I'm not even going to read all that because it's just the opposite of this one. After childbirth, high levels of prolactin induce lactation in mother. During nursing, infant suckling triggers the release of more prolactin, which, boop, D. That is the correct answer. I literally gave you that example in class. That's why examples are important, by the way. You see them all the time. The same ones, over and over again. Let's see. If somebody from Khan Academy sees this, I'm not making any money off this. I'm just using your questions to educate students. I'm not trying to steal your thunder. Okay. Genetic mutation causes a somatic cell to become cancerous. Okay. I think we know the term somatic cell. I don't think it's going to matter, though. We'll see. But I do know that cancer is the unregulated cell division, which is the following most likely explanation for how the mutation caused this malignant transformation. What do I know about mutations? I know it causes a change in a protein, which is going to cause a change in a function. Mutation created an underactive version of the protein that promotes. See, look at this language. This is the trying to trip you up. Creates an underactive version of a protein that promotes cell division. I know that cancer is too much cell division, right? And so if a cell promotes it and it's underactive, that means it's not going to be promoting it. Not A. Mutation creates an underactive version of protein that promotes apoptosis. B is correct answer. Not even reading C and D because apoptosis is kind of the cancer check, right? And not even reading the other ones. Refuse to read them because they're just going to confuse you. Oh, look at here. Here's an epinephrine pathway. This is going to take 2 copy pastes. So there's the thing that we've seen already. Probably stole that picture from them, frankly. I mean, it literally is their picture. I think Con has good pictures. It's a compliment again. All right. The image above depicts the signal transduction pathway of epinephrine. During one step of the pathway, the enzyme glycogen synthase is phosphorylated. Again, this name is just easy, right? Which inactivates the enzyme. Okay. Okay, so I'm going to stop right there. I'm just going to make myself understand this. What is glycogen synthase doing? It's making glycogen. If it's phosphorylated, it inactivates the enzyme, so it stops making glycogen. What is glycogen? Glycogen is the storage of glucose. I'm going to stop storing glucose if this enzyme is activated. See how I talk myself through that? If a toxin prevents the phosphorylation of glycogen synthase during this pathway, which of the following is most likely to occur when epinephrine comes in contact with the receptor? Okay. If a toxin prevents this from happening, right? It's going to prevent this from happening. Then what is going to happen? That means that if glycogen is not breaking down into glucose, then it's going to stay glycogen, right? It's going to stay here. This isn't happening. It's staying right here. Let's go find that. Nope. That's the opposite. Don't even know what that means. And look, this has nothing to do with anything. It's like just a distractor. Glycogen synth will still occur, but glycogen synthesis will not be affected. See, I'm getting turned around too. If a toxin prevents the phosphorylation of glycogen synthase, and if this happens, I'm looking at it backwards, I think. I'm doing the same thing I'm telling you guys not to do. I think it's C. find out. I might be wrong. No. All right, so I'm going to say why it's C. I'm going to give you their reason. Because it was the second part of C that messed me up, that had me like weird. Because I came to the conclusion that it was the first part of C that messed me up. That glycogen breakdown was still going to occur, but glycogen synthesis would still be happening. I knew that was going to happen based on kind of the way I was thinking about it, but it was the first part. So let's read. This is their explanation. Glycogen phosphorylase is unaffected by the toxin, so glycogen breakdown will occur normally. However, the toxin prevents phosphorylation of glycogen synthase, which that's what I thought. So glycogen synthase will not be inactivated. Instead, glycogen synthase will remain active, so glycogen synthase will not be affected, even if heparin. Okay. Got it right. Okay, here's another one. Information below describes the intermediate steps of that thing. Oh, here's a signal cascade. Intermediate just means middle, right? Middle, that's all it means. So that means there's stuff up here and then there's stuff down here. After MEK is activated, it phosphorylates and activates ERK. ERK goes on to phosphorylate and activate a variety of target molecules, including transcription factors. So we don't know what that is yet. If you know what transcription is, it does help. But you can see that's happening here. That promotes cell growth and cell division. That's all you need to know. Which of the following toxins is most likely to inhibit the phosphorylation of c-Myc? Okay. So let's look at this together before we even go look at ABCD. Phosphorylation of c-Myc requires MEK to be activated and requires ERK to be activated, right? So if there's a toxin that prevents one of these from being activated, that is going to inhibit that from happening. I'm done. Not even reading these other ones. I literally said I said I'm looking for that, and that's the first one that I read, right? Let's do another one together. Ooh. All right. The image below depicts a somatic cell nucleus before DNA replication. Okay. So if it's before DNA replication, where are we? There's only one place we can be, right? If we're DNA replication is S phase, so we're here. We're in G1, right? If the cell undergoes mitosis, which of the following correctly depicts the daughter nuclei that would result? It's literally just going to be two cells that are going to look, they're going to be identical. It's A. Wow. Because it's before, right? It's before DNA replication. And so after DNA replication, how many chromosomes we're going to have? We're going to have 8, right? So before, this is what it looks like. And look, let's go back here. After mitosis, what phase is the cell in? It's in G1. What phase are we in currently in the question? G1. It's A. Let's do one more. This one looks weird. I don't know if this is helpful. Y'all let me know. I think just hearing how I process the questions probably is helpful for people. I don't know. I know it probably bothers y'all that I don't read the rest of the answers, but you know what? That's why I don't sit and stare and wonder. Now, some of them still trip me up, though. During cell-mediated immune response, and say some of y'all are going to read this and be like, oh, no, we didn't talk. You don't have to talk about this. This is just an example. During cell-mediated immune response, killer T cells target and kill infected cells. Process is shown below. It really is a number on that cell. I like how it gives the Wikipedia thing here. During the experiment, researchers introduced a substance that caused the class 1 MHC molecule to break down. Okay, let's look at this. Which of the following best describes how a cell-mediated immune response will be affected? All right, so let's look. What is this class 1 MHC molecule doing? So here's the killer T cell, here's the infected cell. It seems like there has to be a connection in order for this to happen, right? In order for this whole process ABC to happen right here, there has to be a connection between killer T and infected cell. That's what it looks like. Just looking at the picture, you don't have to know anything about immune. You don't have to know anything about major histocompatibility complexes. That's what that stands for, but you don't have to know that. You just need to know that it looks like all this is happening because they're touching each other. And so if you took out this square and they're not able to touch, this isn't going to happen. I'm going to go find that. Killer T cell will still bind. No, it won't. No, it won't. So I've already eliminated A and B. Will no longer bind to the infected cell, and the infected cell will instead be killed by... No. No, it's D, right? It's D. Because these two can't bind because that's what it binds to. And this makes up some weird story, right? This is some weird thing that I'd have to like jump off the bus to believe. And so this is just this is the simplest answer right here, right? D. Since it's not binding, that can't happen. D. Oh, I'm having fun. I'm going to do one more. Okay. Following graph shows how cellular DNA content changes over the... Ooh, I love this question. Love it. Cellular DNA changes over the course of a typical mitotic cell in rapid proliferation. Oh my gosh, all these words. Blah, 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 blah. Typical mitotic cell cycle in rapidly proliferating humid cells. All that means is they're reproducing quickly. All right. And so let's see what's happening here. We have DNA content, right? Okay, good. DNA content over here on the y-axis, time. And so we start with 6 picograms. It doesn't even matter. It could be 6 whatever. And then you go a time, and then you get to 12. What's happening here? The DNA is doubling. It's replicating, right? And then you go for a time, and then whoosh. It's no longer doubled anymore because what happens? The cell divided, right? So I'm just trying to understand this graph before I even try to figure out what they're asking me. And I assure you, and you've heard me say this before, do it every time you see a graph. Tell yourself, what is this telling me? All right. Then, based on the graph, which of the following labeled phases corresponds to S phase? Okay, I have to know 2 things in order to answer this question correctly. First thing I have to know is I have to be able to understand this graph, right? I have to be able to look at this graph and read it. And this is literally telling me DNA is going from 6 to 12. Where does it do that? It does it in B. I have to know what the S phase is, which is replication of the DNA, which is when the DNA is doubling. Where is that? It's B. B is the answer. B is the phase. B, B, B. All right, I'm going to do one more. This one looks hard. Actually, the ones that don't have pictures are harder for me. All right. Mutation causes the extracellular domain of a. So extracellular just means outside the cell. Extracellular domain of a G protein coupled receptor to be misfolded. Okay. We already know where we're going with this, right? To be misfolded. The misfolded G protein coupled receptor is packaged into a vesicle that travels to the cell membrane. Upon reaching the cell membrane, the vesicle deposits the misfolded GPCR, which fuses with the cell membrane and functions as a membrane receptor. Which of the following best predicts what will occur whenever the misfolded GPCR is in the presence of a specific ligand? We already know. It can't receive that specific ligand, right? Because it's been misfolded, it's not shaped right. The intercellular domain of the, will change from inactive to new, will not bind with a specific ligand. B. Oh wait, no. What is, okay, I see now. Extracellular, intracellular. The extracellular domain. Okay. The ligand binding. All right, yeah. So, all right, this is actually, this isn't bad, but it takes a little more because this is extracellular, this is intracellular. That is the throw off right there. The ligand binding domain of the misfolded GPCR will not bind with a specific ligand, right? Does that make sense? Because the extracellular domain, it's the place of extracellular. I'm going with D, though I might get this one wrong. No. So B is the distractor there. Let me zoom back up. B is the distractor there, right? It is the, you know that it's not going to bind because you know that it's misfolded. And let's just see why they say it's incorrect. It is true that a misfolded GPCR will not bind with a specific ligand. However, the ligand binding domain of the GPCR is extracellular, not intracellular. And so it's just that one word. And see, that was one that I would have missed if I would have just been like trucking along, not paying attention. So D is the correct answer. One more, and then this is the last one for real, because it's a feedback question. Which of the following statements about feedback mechanisms is most accurate? And so you probably do have to read them all for this one. I don't know. Negative feedback mechanism return a system to its set point. Interesting way of saying that. Yes, that is the case. Because, it's like the hot and cold person. I like A, but because it's like these terms most accurate, I'm gonna, even though everything in me wants to just go and select A and hear that celebration music, I'm gonna go ahead and read the rest of these and explain why they're not true. Positive feedback mechanism bring a system closer to it. No, that's not what? No, that word's just, this isn't a helpful word. Negative feedback mechanisms move a system. No. Further. No. Positive feedback mechanisms maintain a system at its set point. They don't. They amplify something, right? And so A is the correct answer. All right, then. That was good. Hey, any more questions? You guys have anything else you want me to go over or look at? Anything else? That was a good question. That was actually, oh. What is this? They got like level ups and stuff in here? Like XPs? I skipped one of the questions because it didn't have a picture and it like dinged me for it. Probably would have got that business right. Next up is Unit 5. I ain't even trying to do that right now. All right. Well, this has been fun. If y'all don't have any other questions, I'm going to sign off for the evening. I'll be at the school 7:30 in the morning if you want to come see me. Please do so.